50 years ago this spring, two scientists in England bolted together some brass parts and discovered the secret of life.
These were unlikely characters to be working on such an important problem.
The directions for a human being are written in code, 3 billion letters long. These instructions tell our bodies how to live, how to grow, how to die. Researchers call this code the sequence.
Welcome to Secrets of the Sequence. I'm Lucky Severson. Imagine you are tipping back a beer at an English pub when two rather wild-looking characters charge in and announce loudly that they have discovered the secret of life. What would you think? Monty Python? Time to water the whiskey to move on to a quieter pub? It's hard to imagine now what the pub patrons 50 years ago thought when two scientists, Francis Crick and James Watson, announced to the lunch crowd that they had made what is now considered the 20th century's most profound discovery. This year marks the Jubilee anniversary of the discovery of the double helix, the twisted ladder of DNA molecules at the heart of all life on Earth.
When Crick and Watson came bounding out that February day and went to their favorite pub, the Eagle, and they told people over drinks, "We've just found the secret of life," they weren't really exaggerating. It was the secret of life.
What started with Crick and Watson's excited bar talk at the Eagle pub in Cambridge changed the world. Now we hardly blink when we see that DNA manipulation can make a mouse bald and make its offspring glow in the dark. DNA manipulation can allow plants to kill the pests that used to kill them. It can turn goat's milk into spider webs. It is now commonplace to see DNA evidence analyzed in crime investigations. Without an understanding of the double helix, we would not have had Dolly, the first sheep born without a father. And without the helix, Raelians could not have conceived their claims of the first human clone. The suit that Rael wears looks familiar. It calls up images from 1950 science fiction. By mid-century, the advances in the field of microbiology seemed to border on science fiction. In the post-World War II peacetime expansion, science was breaking loose from a narrow focus on military needs, the killing machines, and beginning to explore the basis of life. Electron microscopes were revealing the deep secrets of molecular structures in the nucleus of cells. The fact that deoxyribonucleic acid, DNA, carried genetic information was known, but few thought it was a central mechanism in heredity.
All the big players ended up getting things wrong. It was folks coming out of left field. The notion that DNA was the hereditary material itself, that was heretical.
So there were just a few people who felt that...
Well, through the 1920s and 30s, most smart money was on proteins. Proteins had all these different shapes and could clearly do so many things. They had to be the source of heredity.
Among the few contrarians were Francis Crick and James Watson. Both were working at the time on other research projects and not officially assigned to the study of DNA.
Crick and Watson working on the structure of DNA, these were hardly the orthodox scientific leaders. Jim had come from Indiana, where he was really trained as a zoologist, interested in bird watching, and seemed like an unlikely character to be doing this at the age of 25. And Francis Crick, a retread physicist who had worked in the Admiralty in World War II. These were unlikely characters to be working on such an important problem.
The smart money was on Linus Pauling, a charismatic professor at Caltech in Pasadena, and the leading authority on the structure of protein molecules.
Well, in fact, Linus Pauling published first. He completely scooped Crick and Watson by publishing the structure of DNA before they did, except it turned out to be wrong.
Pauling theorized that DNA took the form of a triple helix radiating around a sugar phosphate central backbone.
And Crick and Watson knew right off the bat that that was nuts, because this sugar phosphate backbone is very negatively charged. You couldn't stick three negatively charged things in pointing at each other. They'd repel each other. How could Linus Pauling blow it like that?
Linus Pauling's error in basic chemistry gave Crick and Watson a break. A few more months in the race to create a theoretical model of the DNA structure. Key to their final success was a technology called X-ray crystallography. It could render the invisible world of molecules visible, but only to those few who could divine what the strange patterns meant. One of the best at using this extraordinarily difficult imaging science was Rosalind Franklin.
You can work out the shape of particular proteins and the shape of DNA, for example. And that's what Rosalind Franklin did. She took crystals of DNA, dried down DNA, shot X-rays into them, got photographic images. And those photographic images, to a practiced eye, like Francis Crick's, immediately told you that DNA was a helix. And that was...
It's a major discovery.
Oh, it's a major thing.
In a few days of intense work, Crick and Watson assembled the first rickety model of the double helix. The model was crucial to the two scientists because it allowed them to see and measure the three-dimensional relationship between the two chains of nucleotides, and thus determine if the theoretical structure would prove scientifically valid, and at the same time agree with what had been revealed in the X-ray patterns.
Rosalind Franklin's pictures of DNA, that is X-ray pictures, which revealed to the well-experienced eye of Francis Crick that DNA had to have a helical structure. Crick had been studying crystallography, and he recognized the helix when he saw it, was just an absolutely crucial part. But Crick and Watson, sitting alone without real data, would not have been able to figure out the structure of the double helix. And it's very clear that Rosalind Franklin, sitting alone looking at the structure of the X-ray crystal she had produced, didn't attempt to build models around it. So while science often seems like the work of the great individual, it is perhaps more than anything the work of an extraordinary community that both cooperates and competes at the same time.
In the half-century since, the 3D model of the double helix has been greatly refined and explored. Now, for the first time in experiments at the Molecular Biology Lab at the University of Geneva, the three-dimensional model has been expanded, adding a fourth dimension, time. The result? A fascinating image that reveals a dynamic living helix far more complex than a simple string of code.
It's a tremendously important, tremendously exciting chase about things, secrets of the world, and you want to know them first. It's a chill. I think most people have never had the experience of being the only person in the world to know some fact about nature.
So the key to our genes may lie not in where they sit, but in how they dance. Biology's complexity seems to grow exponentially as science digs deeper. It seems the more we learn, the more there is to learn. And it poses a question whether it will ever be possible to know it all. That, too, is a secret of life.
I was giving a seminar, I think it was over in Oxford, and sort of briefly mentioned some of these patterns. And there was a very good friend of mine, Nick Proudfoot, came up afterwards. He said, well, that's interesting, a bit like fingerprints, aren't they? And that's it. Let's call them DNA fingerprints. And that actually was a very wise thing to do. If we had called these, say, individually discriminating southern blots mini-satellite hybridization profiles, I mean, we would have killed the technology, absolutely stone dead. Never got off the starting block. But by calling it DNA fingerprints, the public, and we did get quite a lot of press coverage on this, the public sort of, they could twig what we're trying to say. That first murky, horrible-looking DNA fingerprint, that came out of a developing tank at whatever, 9 o'clock in the morning on a Monday, and it was really, I mean, that was a magic moment. I mean, I think my first reaction to looking at the film was, oh, this is a bit of a murky mess. But then I stopped and thought, wait a minute, we've actually got some fantastically variable patterns here. I don't think the term DNA fingerprinting had actually come into my mind then, but we saw the potential for individualization. So first reaction thought, well, first of all, my God, we've solved a very different problem here. Not only have we got access to these really good genetic markers for mapping genes and chromosomes and so on, but we've got a method of biological identification. And so we immediately started thinking of forensics. Could you use this in criminal investigations? Could DNA survive in crime scene samples? Could you match samples with suspects and so on? So those ideas came into mind very quickly. Then we spotted from a little family group on that first autoradiograph that the pattern seemed to be fairly simply inherited, as far as one could tell, with a child made up of half of mum's bands and half of dad's. So that immediately said, if that's true, we've got a method possibly of establishing family relationships. And that meant paternity disputes, which traditionally had always been sort of analysed by blood group testing, and those were good systems. But you can see here, this is going to be something a lot more powerful and definitive. Now, it was a really tough case. Right. First, we didn't have the father for testing. So we had the mother and the boy in dispute. Secondly, we didn't have the sisters for testing over in Ghana. Third, the mother wasn't terribly sure who the true father of this boy was anyway. Apparently there are two fathers, neither of whom were available. So this looked like mission impossible for us. However, what we did have were 3 undisputed children in the family. So what we could do was to take the mother, and these are all done on blood DNA. So we had mother's blood, 3 undisputed children, and you could take mum's DNA fingerprint and the DNA fingerprint of the 3 undisputed kids and reconstruct the DNA fingerprint of the missing father by identifying paternal characters in the children not present in mum. So we now had DNA fingerprint of the missing father, DNA fingerprint of the mother, and then the DNA fingerprint of the boy in dispute, and every single genetic character in that boy matched either mum or the character in the missing father.
What's new, very new, is our ability to manipulate DNA. Today, we can create clones, perfect genetic replicas of mammals from single cells. We can even move DNA from one animal to another, creating designer cows, like Rosie here, capable of producing human milk. Genetic manipulation offers great promise for people suffering from a variety of illnesses, including heart disease. Scientists have discovered that one gene, VEGF2, helps the body grow its own bypass and may one day make bypass surgery unnecessary. And like that one-celled ancestor, we've only just begun. By the year 2003, scientists will have mapped our entire DNA code, identifying all of the estimated 80,000 genes that make us human. No telling what we can do with that information. Typically, we think of sheep as followers. But then along came Dolly. This little lamb led the way to an enormous breakthrough in the science of cloning. In normal sexual reproduction, genetic information carried in chromosomes from 2 parents combine to form a fertilized egg. The blue packets of information on the left represent genes from a male, on the right, a female. When their chromosomes combine in the center, the cell begins to divide and create a unique individual. Cloning creates perfect genetic reproductions of a single animal. Dolly is the first mammal ever cloned. In 1997, scientists produced her using a single cell from an adult sheep. Most scientists used to believe that using a cell from an adult animal to create a copy would be impossible. As cells age, they specialize, becoming a particular type of cell, like a skin cell or a nerve cell. Instructions for creating other life are switched off. Dolly proved them wrong. Dolly's creators collected cells from an adult sheep and starved them of certain nutrients. The process essentially backed up the cell's development. Losing their specialized instructions, the cells were able to create new and different life. One cell went on to become Dolly. Other cloning techniques have been developed. One, designed to make herds of cows, takes place in a lab that's kept as hot as a cow's womb, 110 degrees Fahrenheit. Using the eyes of a powerful microscope, scientists remove the nucleus of an egg cell, leaving the egg with no genetic directions. Then, they add the DNA from the cow they wish to clone. New instructions and a jolt of electricity start a new life. But the cow clones develop the old-fashioned way, inside a female. Still, only 1 in 95 of these engineered pregnancies produces a calf. So why go to all the trouble? To preserve life's infinite variety is one answer. Few more than 1,000 giant pandas still exist in the wilds of China, and cloning just might be a way to spare them from extinction. Scientists have already gathered DNA from a giant panda and plan to produce a panda clone. For a long time, scientists have known that genes control the way we look. More recently, they've jumped at the notion that genes may influence behavior, even risk-taking.
Yeah, I guess I am a risk taker. I mean, if someone dares me to do something, I gotta do it. You know, I can't say, no, I'm not gonna do that. My younger brother is definitely a dare taker. I mean, I think he's worse than I am. And I guess we both get it from my mom.
But geneticist Dr. Dean Hamer cautions that genes don't account for specific behaviors.
All that genes do is to code for proteins, and all the proteins do is to control chemical reactions inside the brain. So there is no gene that says go bungee jumping or jump off the high dive or smoke a cigarette. The genes simply alter our brain chemistry in rather subtle ways.
So just what are those ways? Risk takers may have what Hamer calls a novelty gene. New experiences will release a chemical called dopamine, making the experience a pleasure. People who prefer to keep their feet on the ground may have an anxiety gene that releases serotonin instead of dopamine. This chemical response to new challenges makes people feel a little anxious, even worried. Ready to take the plunge? Genes don't make choices for us, but they clearly influence the way our choices make us feel. DNA is present at every crime scene, just waiting to tell its secrets. Because DNA is found in each and every part of us, it's more likely to be left behind than fingerprints. Detectives collect DNA from blood, hair, even the saliva left on the back of old postage stamps. Gathered samples are X-rayed and analyzed. The black bands show the pattern of chemicals that make up the rungs of an individual's ladder of DNA. This sample was taken from a violent crime scene. And this sample came from a suspect. If the two match, the suspect will most likely become the accused. Unless a sample's contaminated, DNA doesn't lie. Collecting DNA from crime scenes is now standard police procedure, and the FBI considers DNA its best crime-solving tool. In 1994, a new software program helped law enforcement agencies share DNA evidence. Called CODIS, the program compares samples taken from a crime scene with any and all other samples stored in the system. Criminals are turned in by their own genes. When the computer finds a match, it's called a cold hit. Already, CODIS has solved over 300 crimes. Even old, unsolved crimes can be reopened with DNA evidence. In 1954, the murder of Marilyn Shepard put her husband, Dr. Sam Shepard, behind bars. Convinced of his father's innocence, Shepard's son pursued the case even after Sam Shepard died. DNA tests proved that Sam Shepard's defense had been the truth. Someone else had entered his home and murdered Marilyn. DNA taken from blood at the crime scene did not match Shepard's or his wife's.
To tell you the truth, I've never once ever thought that I was a creator. I feel more like a manipulator of taking a very complex, a very beautiful system and manipulating it in ways that it can be of benefit to mankind.
Dr. James Roble may not think of himself as a creator, but he is. He and other geneticists are designing new animals by adding the genes of one to another. The man-made creatures are known as transgenics. These transgenics are cows outfitted with a human gene. Human proteins produced in their milk are the same proteins needed to create medicines for burn victims and patients afflicted with cystic fibrosis and hemophilia. Clone these customized cows, and you have a drug factory on the hoof.
We can get literally billions of these cells, and we can do all sorts of things with those in culture. And you look at the petri dish full of these, 100 million cells, and you think, gee, this is quite a large herd of cattle here, because we can theoretically turn every one of those into a cow.
Meet Rosie. She's a transgenic model equipped with yet another human gene. Her creators expect cows of her design to produce human milk. A dairy farm producing hundreds of gallons could help nourish starving children or babies born prematurely. Transgenic pigs may be a solution for people in need of organ transplants. This pig is being surgically implanted with genetically altered embryos, designed to grow hearts compatible with our own. If all this genetic mixing and matching strikes you as risky, rest assured, it is. The dangers are ethical as well as medical. Is it right to turn one species into a processing plant for another? Will new viruses come with new genes? And breakthrough technologies are coming faster than answers. Still, risky business is booming.